Hello! In this video I'm going to be taking you through the CSF and the spinal cord. I'm going to start with the anatomy of the ventricular system. So firstly we have here our lateral ventricles. The lateral ventricles have an anterior horn, an occipital horn which sits posteriorly, and then this is the main body of the lateral ventricle. And then in the temporal lobe we have our temporal horn which comes down here. The lateral ventricles then drain into our third ventricle via our IV foramen or our foramen of Monro, and this here centrally is our third ventricle. This drains through our cerebral aqueduct into our fourth ventricle, and then our fourth ventricle can either drain into the subarachnoid space or it can drain straight down into our central canal of our spinal cord. So firstly our lateral ventricles, which we can see here on our images. So superiorly here we have our anterior horn and the main body, and as we come a level lower we can see this main body moving back towards our occipital horns in the posterior occipital lobe, and then you can see an extension of these here. And then as we go another level lower you can see that these have now moved round to be in the temporal lobe, and this is our temporal horn. Now we also have our foramen of Monro, which is how our lateral ventricles drain into our third ventricle, but these can't be seen on the imaging, and our septum pellucidum, which we can see in the middle here, which separates our two lateral ventricles. Our third ventricle is entered via the foramen of Monro from our lateral ventricles, and it sits between the two thalami. So we have a thalamus on either side, which are connected in the middle by an interthalamic adhesion. And our third ventricle sits in this gap between the two, and then is interrupted in the middle by this interthalamic adhesion. The third ventricle then drains through our cerebral aqueduct into our fourth ventricle. So our fourth ventricle sits behind the pons. So this is why the posterior wall of the pons is sometimes referred to as the floor of our fourth ventricle. And this means that problems within the fourth ventricle can lead to compression of the pons. And then we drain from our fourth ventricle either into the subarachnoid space, and this is via our aperture of Magendie or our apertures of Luschke. So you can remember that the aperture of Magendie is in the midline and it's medial because it begins with an M, and then our apertures of Luschke, which we have two of, sit laterally. So we have a left and right aperture of Luschke, which drain into our subarachnoid space. We also can continue straight down from our fourth ventricle into the central canal of the spinal cord, which also contains CSF. So this is the journey of our CSF. So it's first produced by ependymal cells of the choroid plexus, and the choroid plexus surrounds the whole of our ventricular system, and these ependymal cells are how the CSF gets produced. So for the sake of this, I'm going to start with the lateral ventricles, but this CSF can be produced in the third or fourth ventricle. So from our lateral ventricles, we then travel through our foramen of Monro to our third ventricle, where we travel through our cerebral aqueduct to our fourth ventricle. And then our fourth ventricle can drain into our subarachnoid space via our apertures of Luschke and Magendie, and then from our subarachnoid space, or indeed from our central canal of our spinal cord, the CSF then goes into our venous blood via these arachnoid granulations. So now we move on to hydrocephalus, which is a problem with the CSF, and literally means fluid drain. So we have two different types of hydrocephalus. The first is obstructive, and this, unsurprisingly, is when there's an obstruction within the ventricular system, and this means that the CSF can't flow effectively but is still being produced, and so you have this excess of fluid. And you often think of hydrocephalus in neonates or young children, and because the bones of their skull have yet to fuse together, they are able to expand. So when they have this hydrocephalus, the heads expand and you get these very large swollen heads. And things that can cause this are cysts, tumours, or congenital defects such as cerebral aqueduct stenosis. So if that's too narrow, then there won't be a good flow of CSF. We also have our communicating hydrocephalus, and this is where the CSF is able to get into the subarachnoid space, but from there it's not absorbed properly by these arachnoid granulations. So this can be caused by things like a subarachnoid hemorrhage or by meningitis, which cause problems in the arachnoid mater and therefore mean that there isn't good absorption from the CSF. So we also have our cisterns, and these are areas of the subarachnoid space which is widened, so there's a large gathering of CSF in this area, and that's called a cistern of CSF. So we can use these when we're looking at imaging, as we can see if they've changed shape or size, that there's likely to be a raised intracranial pressure or midline shift caused by maybe a bleed or a tumour or something like that. So the cisterns we have are our quadrigeminal cistern, which sits just here, anterosuperior to our cerebellum, our cisterna magna, which sits below our cerebellum just here, which is next to our foramen magnum that our brainstem and spinal cord pass through. We have our interpeduncular cistern, which sits just between our cerebral peduncles, which sit at the anterior part of our midbrain. So that's just here. And we have our prepontine cistern, which sits just anterior to our pons here. And finally, we have our lumbar cistern, and this is inferior to the terminalization of our spinal cord. And this means that we have a large area of CSF below our spinal cord, and this is where we try and access when we do a lumbar puncture. So next I'm going to talk about the blood-brain barrier, which can be split into two parts. We have our blood ECF barrier, which is the main part between the blood and our neural tissue. And so this is sometimes what people are referring to when they say blood-brain barrier. But we also have the secondary part called the blood CSF barrier, which is the part between our ventricular system containing our CSF and our blood. So this has a number of functions, including controlling substrates and molecules that can move between the blood and the CSF and the neural tissue. They control ion movement, as that allows you to maintain a action potential and resting potential. And it means that neurotransmitters can't pass through into the neural tissue and affect how things are working within the brain. So first I'll talk about the blood CSF barrier. So surrounding our ventricular system, as we've said, we have this choroid plexus where the ependymal cells produce our CSF. But we also have these epithelial cells. And these epithelial cells have tight junctions between them. And this means that the blood here, which is running between the neural tissue and our ventricle, is unable to pass directly through into the ventricle. And these epithelial cells have microvilli and mitochondria and special transporters, allowing them to pass through certain things such as glucose, sodium and chloride, and absorbing some of the waste and excess solutes that are within the ventricle. But it means that other things that are contained within the blood, which don't have these special transporter systems, are unable to pass through. We also have the blood ECF barrier, and this is between our capillaries and our neural tissue. And again, we have these tight junctions between the endothelium of our capillary. And this means, again, that ions and molecules and our neurotransmitters aren't able to cross this barrier. And they must again cross these endothelial cell membranes, and you have specific transporters that allow only certain things to get past. And these are sometimes targets for new drugs. So the final thing I'm going to talk about is the anatomy of our spinal cord. So if we take a slice of our spinal cord, we can have a look at it in a few different areas. So we have our white matter and our grey matter. In our white matter, we have 3 funiculi, anterior, lateral, and posterior. And these are essentially the areas of our white matter. And then we have this anterior white commissure here, which is essentially the crossing between the two sides. In our grey matter, we have our dorsal horn, which is where our sensory fibres enter. We have our lateral horn, which is where we have autonomic fibres synapsing, and this only happens where we have autonomic fibres entering the spinal cord. So this is T1 to L2 and S2 to S4. That's T1 to L2 through our sympathetic and S2 to S4 through our parasympathetic. And then we also have this ventral anterior grey horn, which is where our motor fibres enter and synapse. We also have, as I've been saying throughout the video, this central canal of the spinal cord, which runs through the middle and contains CSF. So the reason that this is of interest is that you can get a syrinx forming, which is sort of like a cyst in this central canal, and this can compress on the anterior white commissure. And this is called syringomyelia, and this can have various effects, which will be discussed in the video on ascending and descending tracts. So at the bottom of our spinal cord, we have a number of landmarks. So we know that L1 and L2 is about where the adult spinal cord normally terminates. But in a child and at birth, it's around L3 to L4. And then we have below this level of termination of our spinal cord, this lumbar cistern that we were saying about earlier, that we want to access when we do a lumbar puncture. And this ends down at S2. So this means we have a nice big space we can access. So if you refer back to your block 1 surface anatomy, then you'll remember that we have our transpyloric plane at L1, which is about where our spinal cord terminates. And then we have our subcostal plane at L3, which is about where it terminates at birth, and that's at the bottom of our costal margin. And then at L4, we have our supracristal plane, and that is the top of our iliac crests. And then we have the transtubercular plane at L5. So if we want to perform a lumbar puncture, we obviously want to go below the level of our spinal cord at L1, L2. So we'd normally go at L3, L4, or L4, L5. And the most common way to do this is to identify the iliac crest and then draw a line back. And this should give you the L4 vertebrae. And then you can use the gap just below that, and that will give you your L4, L5 space. And you can be confident that you're below the level of the termination of our spinal cord. So when you do a lumbar puncture, you'll go through a number of layers. And this is a recap of the ligaments of the spine that you pass through. So you first have your skin and your connective tissue that lies below that. You then have your supraspinous ligament, which passes all the way along the posterior part of the vertebral column. And then you have your interspinous ligament. And there's an interspinous ligament between the spines of each of the vertebrae. You then have your ligamentum flavum, and as you pass through this, there'll be it's sometimes described as a pop, but it's more like a give, where you'll feel that it's slightly easier to start passing through because it's quite firm. You then will pass through your epidural fat and then your duromator. And this will again give a pop or give as you pass through it. And then you pass through your arachnoid mater. But it's worth noting that your duromator and arachnoid mater are typically very closely adhered to each other unless there's a problem. So when you feel this second pop or give, then that's when you know you're now in your subarachnoid space. And this is obviously where our CSF sits. So this is where you want to take your sample of CSF from. So the times when you do a lumbar puncture include if you want to give spinal anesthesia or an epidural, but obviously for an epidural you'd stop in once you've felt that first pop, as that would mean you're in the epidural space. And you do it if you suspected meningitis and wanted to take a sample to see if there were any bacteria or viruses within the CSF, or if you wanted to suspected a subarachnoid hemorrhage, in which case when you do this you may find that there's some blood cells within the CSF. You can also do it to measure the intracranial pressure, as this will give you an idea of how much force is being put on the CSF that's within the subarachnoid space in the spinal cord. So that's everything you need to know about the CSF and about your spinal cord. So thank you very much for listening.